Earlier this year, we reached a point where we could do millions of cells in a single experiment, but from a relatively smaller number of different specimens. So one mouse, or one heart, or one tumor. Now, with sci-Plex, we can do millions of cells from thousands or even tens of thousands of different specimens.
It's a method we call sci-Plex. It's a way to perform single-cell sequencing while linking it to multiple perturbations. And in this case, we scale that to do thousands of perturbations at a time. Current ways that people test drugs include kind of a more specific life or death kind of scenario. So they'll screen for compounds that kill the cell of interest. There's more specific versions of that where researchers will look for drugs that actually inhibit a specific protein. This technology actually occupies a niche between those two, where you can get a kind of global view of what the cell is doing in response to a drug. So we think it's going to be really powerful to actually categorize drugs and say what their mechanism of action is.
We give many different people the same handful of drugs, and it works for some and doesn't for others. And this potentially could help us understand, sci-Plex could potentially help us understand why that is.
The way that drug discovery is done right now, we just know whether a drug can kill a cell or not. We don't necessarily know its mechanism of action. So we anticipate that this technology can be used to uncover the mechanism of action of different drugs and potentially answer why certain drugs become resistant to certain compounds, or which sets of cells become resistant to certain compounds.
I think that sci-Plex could be a very useful tool for precision medicine because ultimately, when someone gets sick, like with cancer, we want to kill the whole tumor. We don't want to kill part of the tumor. We don't want to kill some of the cells. We want to kill all the cells. So understanding cellular heterogeneity and why some individual cells respond one way to a drug and others respond a different way, understanding those differences is critical to designing therapies that will be completely effective.
Our hope is that using single-cell sequencing and technology like sci-Plex, you can actually uncover what makes those cells that live different from those cells that die.
I'm really interested to see how the single-cell genomics community sort of picks this up and uses it for things that we did not anticipate, because that is happening all the time in our field. People are, technology developers and experimental biologists are repurposing techniques for all kinds of things that, that the original developers did not envision.